Hello, and thank you for joining us today. Welcome to our webinar entitled Targeting DNA Damage Repair for Drug Discovery Using the Transcreener ADPR Assay. I'm Justin Brink, COO here at Bellbrook Labs. Please note that if you have any questions during the webinar, you can type them into the Q&A section, not in the chat box, please. We'll do our best to answer them during the live discussion. I'd like to introduce to you our presenter, Ha Pham, Senior Scientist at Bellbrook. Ha has a PhD in biochemistry from the University of Illinois Chicago, where she studied and developed a variety of different assay development techniques. Ha was instrumental in developing these assays, and I am excited to hear about her work, which she will share with us now. Thank you, and with that, here's Ha.
Thank you, Justin, and thank you everybody for being here. So today's topic gonna be targeting DNA damage repair for drug discovery using the Transcreener ADPR assay. So this is the agenda for today. First, I will talk about ADPR producing enzyme and how it's important for drug discovery. Then I'm gonna talk about how we develop the Transcreener ADPR assay and how we applied it to the specific case for CD38 and PARG. Finally, I'm gonna address any question you have. So if you have any question during the webinar, please type in the Q&A and we're gonna address them all at the end of the presentation. All right, so let's dive in the first part. So a little introduction about ADP homeostasis. So ADP-ribosylation is a posttranslational modification. So for proteins, the ADP-ribosylation gonna add ADPR or poly-ADPR on different amino acids of the protein. And this is a reversible process. As you can see, the enzyme that remove the addition. So at it adding or removing ADPR, it turns on and off specific pathway, and therefore it controls important cellular processes, such as DNA damage repair, stress, and immune responses. So to maintain the cellular homeostasis of ADPR, there are many enzymes that get involved. For example, we have transferases, we have hydrolases, the ones that add or remove the ADPR or poly-ADPR. So in this case, we have PARP1, we have PARG to remove the poly-ADPR or ARH3, ARH1. We also have enzymes that control the NAD level. So NAD level is an important material for ADP-ribosylation. So therefore, the enzyme get involved in this process as well. So those are NADAs, such as CD38. So for specific topic today, we're going to focus on the CD38 and the PARG. So about CD38, it's a cluster of differentiation 38. It degrades NAD to produce ADPR. And then for a specific pathway, this ADPR is chopped down to AMP by ENPP1, and then chopped down to adenosine by CD73. This adenosine acts as a trigger for immune suppression in tumor microenvironment, and that allows the cancer to grow. So as a result, if we inhibit the CD38, it can be a way for cancer therapy for many cancers, such as lung or prostate cancer. And by the way, we have assay for ENPP1, and also we have assay for CD73 as well. So if you want to target the entire pathway, so please check our website for further information. And for the case of PARG, it is Poly-ADP-ribose glycohydrolase. It works together with PARP1. So as DNA got damaged, PARP gonna synthesize poly-ADPR on itself or on other protein. And PARG gonna act as a reverse process by removing the poly-ADPR from PARP1 and then further chop it down into ADPR. So that helps to recycle the PARP1 or negatively regulate the process. As a result, if we inhibit the PARG, we can disrupt the normal process of DNA damage repair, and therefore it can be potential as a tumor or anti-tumor drugs. So here Bell Brooks come in. We want to aid drug discovery for those ADPR producing enzyme. So we develop this assay, the Transcreener ADPR assay. And here I want to talk about how the assay works. So as the target enzyme produces ADPR, we use a coupling enzyme to convert ADPR to AMP for detection. So this AMP gonna displace the tracer from the AMP antibody, and that's gonna result in a decrease in AP. So as you can see, this is a far red AP readout, and it help to minimize compound interferences with the far red tracer, the Alexa Fluor 633 tracer. So how this assay work, how we run this assay. So this is an example of a 384 well plate. We're gonna have 10 microliter of enzyme reaction, and in this 10 microliter enzyme reaction, we have the coupling enzyme in there to convert ADPR to AMP in real time. And in a little bit, I'm going to share with you how we determine the amount of coupling enzyme because it works in real time. The excess amount of coupling enzyme is important. And after incubating at certain temperature and time, we add 10 microliter of detection mix. In this detection mix, we have the antibody, the tracer, and a stop buffer to stop the coupling enzyme. So as a result, the Transcreener ADPR assay will be endpoint assay for any target enzyme because it stops the coupling enzyme, so no more detection, even though if the target enzyme is still working on the background. So overall, this assay is a homogeneous assay with very simple mix and read format. And we use a tracer at the far right so that we can minimize compound interferences. And here how we determine the amount of coupling enzyme. And it works in real time. It's important that it's not the rate limiting step during the enzyme reaction. That means like we have to get coupling enzyme in a lot of access so that we can readily convert all the ADP to AMP in real time. In order to do that, I do the target enzyme titration for each target enzyme with different amount of coupling enzymes, and then I convert the raw data into ADPR form, and then I use that to calculate the rate of the enzyme. So the amount of coupling enzyme that doesn't change the rate of the target enzyme is the amount that I go for. And then I take that amount and multiply with 5 to get the final working concentration for the coupling enzyme. So why is that important? So the limitation of a coupling enzyme assay is a false positive result for an inhibitor. That means the inhibitor may inhibit the coupling enzyme and it results in a dose response curve. So when we provide the enzyme in 5 times access, let's say if an inhibitor inhibits the coupling enzyme for 80%, we still have enough coupling enzyme to convert all the ADPR to AMP for detection. And that way to minimize the potential assay interference that may inhibit with the coupling enzyme. And here is the sensitivity and the selectivity of our assay. So the first graph shows the standard curve, ADPR, different amount, and then the MP respond to that. And then we have this standard curve, a different amount of antibody. So as you can see, this assay can detect at low at 10 nanomolar of the ADPR with a positive Z prime, and then up to 50 micromolar ADPR. And then one cool thing about this assay is we can tune the dynamic range by adjusting the amount of antibody used and antibody concentration. So for example here, if you want to detect from 10 nanomolar to about 1 micromolar, you can use 2 microgram per ml antibody. And if somebody wants to use to detect from 1 micromolar of ADPR and up to 10 micromolar of ADPR, they can use 50 microgram per ml of the antibody. So that will fit to your application if you want to use a lot of substrate or what type of amount of enzyme that you use. You can tune your assay towards that. And the second graph, I want to tell about the selectivity of the assay. Before going to this graph, I would like to say the AMP2 and GMP2 antibody is well validated antibody in our AMP2, GMP2 transcreener assay. So this antibody have very high selectivity for AMP compared to ADP or ATP, like more than several thousand-fold different for selectivity between AMP, ADP, or ATP. And then for the coupling enzyme, as you can see, it have very specific activity toward ADPR compared to similar nucleotides such as ADPR or NAD in as shown here. So overall, this assay is very selective for detecting the ADPR. So now let's dive in the second part of my presentation, where I'm gonna tell you about how I apply the transcreener ADPR to specific case of CD38 and also for the PARG. So before that, I want to give you a little introduction about the CD38 assay system. So recently, we make a lot of assay system with the idea that when you have this assay system, you have everything to run your enzyme reaction. And then it works together with the Transcreener ADPR assay so that this provides you everything you need for detection. So the idea is with these two kits, you have everything ready to run screening or to run dose response curve. All you need is a plate reader and some pipettes. So let's say, for example, in CD38 assay system, we're going to provide the enzyme and also the NAD substrate, and then we have CD38 assay buffer, and we have a 384 well plate to run the enzyme reaction. And after the enzyme reaction is done, then you can use the ADPR assay kit where we have everything for detection, the antibody, the chaser, the coupling enzyme, stop buffer to stop the assay, and then ADPR to produce the standard curve. So really, I mean, these two kits, you have everything to run the assay from material down to protocols to run the thing that you need for your applications. So this is how CD38 assay system data look like. So here I have CD38 enzyme titration, and then in the middle is the standard curve, and the last one is the interpolated data where it shows the ADPR form versus the amount of CD38 here. So before going into its graph, I want to talk about the assay condition. So I really work to optimize everything in the assay reaction and also to optimize what to use for the detection here. So for a 384 well plate, we use 10 microliter reaction. And if you plan to use other plate for your experiment, please adjust the volume accordingly. So in the 10 microliter reaction, we have NAD plus, which is a substrate, at 15 micromolar, which is a low-end reported km for NAD plus. And then we have the coupling enzyme. We provided 400x, and the working concentration gonna be 1x. And then we have the CD38 assay buffer. So after adding this to the enzyme and incubate for 60 minutes at 30 C, you're gonna add 10 microliter of stop and detection mix. So the final concentration, the final volume for your assay will be 20 microliter for 384 well plates. So in 10 microliter stop and detection mix, we have the antibody concentration already optimized for you, which is at 10 microgram per ml. And then we have the chaser at 8 nanomolar and stop buffer B. So this stop buffer, in this case, it's gonna stop both the coupling enzyme and the CD38. Okay, so one question for the data, why you see the sigmoidal curve for an enzyme titration? So this is a competition assay, and at the binding occur, we always have a sigmoidal curve for the enzyme titration. And then if you use a ADPR standard curve to convert the raw MP value to the amount of ADPR form, we're gonna have a linear relationship between product form and the amount of enzyme as expected. And then the second question is from here, how you gonna decide what amount of enzyme for your application. So my rules of thumb is you're gonna choose the amount of enzyme so that you have at least about 100 MP shifts. So that way you make sure you have an excellent data quality and robust ZPRAM. And then the second rule is you're gonna choose amount of enzyme so that you are in the initial velocity. That means less than 10 to 20% substrate conversion. So in this case, for this specific case, I would choose about 16 picomolar CD38 because that will give me at least 150 MP shift from here to here. A good range to work with. And then I have about 1.7 micromolar ADPR form, and that is about 13% conversion from the 15 micromolar NAT that I use as a substrate. And then my final question here is, do you have to do this enzyme titration to figure out the amount of enzyme every time? So the answer is because we have all the condition optimized. So for each lot of enzyme, we're going to have the CFA, which we provide with the titration and also the interpolated data. So if you run a standard condition, and you can refer to the CFA to determine the amount of enzyme for your application. But if you decide to go away from the standard condition, for example, you want to run at higher substrate concentration or using different buffers, then I would say you may need to rerun the enzyme titration to determine the amount. Okay, with the standard condition and with 16 picomolar of enzyme, I do a pilot screen of more than 1,000 small molecules of TOCRIS 2.0 library here. So those are my screening data where I have the test compounds, I have the no inhibition control, so that I can use that to determine the line here, which is 3 standard deviation away from the no inhibition control. I have no enzyme control and positive compound control to calculate the statistics for this screening. And one important thing on this side, I show the interference control run together with the pilot screen. So I want to emphasize that it's important to run the interference screen to eliminate any false positive result. So in this interference control screen, I run everything same as the pilot screen, but replace the amount of enzyme by the amount of ADPR produced. In this case, 1.7 micromolar ADPR with 16 picomolar of enzyme used. So as you can see here, I have 1, 2, 3, 4 hit, but out of that 4 hits, 3 of them show some interference with my detection. So I'm not going to go with the 3 hits, and that the 2 hits that I'm going to go follow with dose response curve and further study. So this is a dose response curve for that hit and a positive control. So this hit is HU9516, a CDK2 inhibitor, and in this case, it shows inhibition for CD38 at 1 micromolar IC50, and then the C78C is a positive inhibitor, well known, that I use as a control for my assay, and it shows about 36 picomolar for IC50, similar to the literature and reported values. So the Z-prime for the screening is 0.93, calculate from no enzyme control to the complete reaction, no inhibition control here. And this is very excellent data quality for a high throughput screening assay. So now I would like to talk about PARG assay system. Similar to the CD38, we already optimized condition to run the PARG assay system. A little bit different here. We have Poly-ADPR as the substrate, and then it can be run at km or a subsaturated condition, which is 5 times km. And then we have the coupling enzyme at 1x and assay buffer at 1x. And again, this is condition for 384 well plate, and for other plate types, just adjust the volume accordingly. And then after 60 minutes at 30C, we're going to add the complete to complete the assay with 10 microliter stop and detect buffer. And in this buffer, we have the antibody, the tracer, and stop buffer B. So in this case, stop buffer B only stop the coupling enzyme and not stop the PARG, but it still works as an endpoint assay. So for the amount of antibody, as you can see, we can use 1 or 2 microgram per ml here. So because when I use km, I have less ADPR produced, so I want to make the assay a little more sensitive. So that's why I choose less amount of antibody to tune my assay dynamic to what I want, so that I mentioned before. And with this particular example, I would go with about 6 picomolar of PARG for any screening or dose response curve. About the substrate, this is a polymer substrate produced by chemoenzymatic synthesis, and the polymer size distribution can vary slightly from lot to lot. So please refer to CFA to know the km for a specific lot. And here I want to show an example for PARG inhibitor dose response curve profiling. So we have 3 different kinds of dose response curve here. The MP value, this is PDD, a known inhibitor for PARG, and I use that an example here. And then I have the MP value converted to ADPR form, and then I have the percent inhibition further calculate from the amount ADPR form. So I run at km, also at subsaturated condition 5 times km, and I have the interference control here to show that the inhibition is actually inhibition not about any interfering with the interfering with the detection. As you can see, this is the case here. It's really the inhibition response, not about interference response. So I would say the IC50 values that you obtain from the product formation or percent inhibition would be the most accurate. For IC50, for the MP values, it's close to those value, but it's not the best because it in a dose response curve like that, it also combined with the binding response. So therefore, it doesn't reflect truly the IC50 for the compounds inhibitor that you test. And also, the way that we can detect at km and also at 5 times km substrates is a benefit, because if you want to study an inhibitor and want to know if it's competitive with the substrate, with poly-ADPR, then that can be used. For example, in this case, the PBD is very similar IC50 for the 5 times km versus the km. So therefore, we can say PBD is not a competitive inhibitor. So the Z-prime for PARG assay system, either at 5 times km or at km substrate, is greater than 0.8, so that be very excellent for a high throughput screening assay. And then with that, I would like to go to the summary where I already tell you about the ADPR assay. It's a far-red epi homogeneous assay. It can detect ADPR as low as 10 nanomolar. It can go up to 50 micromolar. And the data quality is excellent. I would say the Z-prime is greater than 0.7 for both two assays. And then the signal quality greater than 100 MP polar shift. And then I have shown you, I do the pilot screen and also the response for profiling for CD38 and for the PARG. And then I really hope that with the Transcreener ADPR assay, it will facilitate the rapid discovery of inhibitor for CD38 and PARG, and also other related ADPR producing enzyme. And with that, I would like to thank you very much for listening, and then I would like to answer any questions that you may have.
Hi, so we have a few questions here. We'll start with this one here. You mentioned tuning the dynamic range for ADPR detection. Is that something we will have to do with CD38 or PARG?
Oh, okay, that's a good question. So because the idea for the assay system kit is we provide you everything really from the material to the protocol. So you just need to pick up a protocol. So in that protocol, I would already adjust and optimize the antibody concentration to use with the standard condition that we provide. So you really don't have to tune the antibody concentration anymore. But if you want to go away from the standard condition, then you may need to adjust the amount of antibody a little bit to what sensitivity that you want.
Another question here. Since the assay uses coupling enzymes, how do you determine if the hit is real or potential interference? What steps would you take to do that?
Okay, I think that's an important thing to know. So when I think not just for the Transcreener, but for any assay, when you run screening, I would say it good to have an interference screening run with your screening. In the interference screening, we just do everything the same as the pilot screen, but omit the enzyme and replace that with the amount of product form. That way we can know if a response is from the inhibitor or just from interfering with the detection, as I did for my screening for CD38 or the response curve with the PARG.
Excellent. So another question here. What plate reader settings do I need to consider for this assay?
Oh, okay. So for plate reader, I would say any plate reader that can perform the AP. The dye that we use for the tracer is 633 Alexa. I believe it's very common dye, so chances are you already have the optics for those dyes. But if not, then you're looking for a filter and optics for exciting the dye at around 575 nanometer, and then emission at around like 633 nanometer.
Great. One more question here. Have you used the assay to successfully calculate enzyme kinetics, for example, the KM? Show a little bit of that. Do you have some guides to help with the assay development?
Oh, so for the enzyme kinetics, I do use the assay to determine the poly-ADPR KM, as I show you in the slides. And then if you want to learn more how the way I do that, probably you can reach out to me at info@bearbrookLab.com. I can give you further protocol for doing that. And then for how to develop the assay, so my approach first, I gotta kind of figure out the enzyme activity first with any literature or anything that I can find, and then I will work from there. First, I'm gonna optimize the assay buffer, pH, the salt, and any kind of detergent there. And after that, I'm gonna optimize the substrate concentration, like figure out the Km and see if the assay run at Km or saturated condition. And then I will go from there to the detection, like what's the best detection to give my Z prime that I want or the MP value that I want. So that's my typical approach for develop an assay.
All right, one more here. Do you offer custom kits to use our own proteins?
Yes, so you can see we have two separate kits. The first one is a CD38 assay system or other assay system where we have enzyme everything for running the enzyme reaction, and the second kit is for detection. So if you have something on your own, you can still get the detection kit and then follow the protocol to see if it works for your target with our detection kit. But I would say you may need to fine-tuning your system a little bit towards the one that you customize for the target that you have.
Well, thank you, Ha. That's all the time we have for today. With any other questions, you can certainly contact Ha or myself. A good email for that is info@bellbrooklabs.com, and someone will definitely take care of you. A copy of this webinar will be sent within the next 24 hours, so everyone can review that. Also, please go to our website, bellbrooklabs.com, for our latest posters on DNA damage response and innate immunity. Thank you, Ha, and thank you all for joining us today. Goodbye.
Goodbye.